Welcome to OmicsBox, bioinformatics made easy.
So the goal of this section is to first understand what pseudotime is and what trajectory inference is, and how you can confidently infer single-cell trajectories. And once you have inferred them, how you can generate biological insights in OmicsBox. So let's first start the discussion with trajectory inference. Now we know that all the cells come from the pre-existing cells, and typically this starts from the progenitor cells or the stem cell-like cells and goes until to the stage where the cell becomes functional or specialized. Now, collectively, all of these stages, they make up a cell lineage, and these cell lineages, they represent basically a biological process. And a biological process can have multiple lineages or some different lineages where a cell can divide into more than one cell type. For example, in this case, there is a linear lineage where one cell type is getting differentiated into a very specialized cell. But there can be cases where you might end up having two different specialized cell states. For example, you can see that in the case of hematopoietic stem cell differentiation, where you end up having different kind of blood cells from the hematopoietic stem cells. So when you perform single cell sequencing, what you capture is these cell stages collectively, which are coming from a biological process, which has a dynamic nature. And the goal of the trajectory inference is to provide order to these cell stages. So you try to put these cell stages in a way that they represent their actual biological process. And to do that, what you do is you first reduce the subspace. For example, you have thousands of genes, so you reduce them to the latent space using techniques like PCA, UMAP, or t-SNE. And then you try to represent the cells in that latent space. And then you try to put the string through all the cells. And this string is known as a trajectory. And the idea is to put the progenitor cells first and the highly differentiated or the functional or specialized cells towards the last. And the intermediate cells, they are represented in the middle. So basically, this is a representation of a simple trajectory where you have a linear process. These are the progenitor cells, and these are the differentiated cells. So once you have them represented in the latent space and you have learned the trajectory, what you do is you assign them pseudotime, which typically starts from 0 for most of the methods. So what pseudotime is? So it's a continuous variable which represents the dynamic nature of the cells, and it's a proxy value for the cell's developmental time. So you can understand it like an age of the cell, and it's not related to the experimental capture time. Rather, it represents a relative measure of progress along the biological process. So the idea is to stratify, provide order, not to quantify. So a pseudotime value is arbitrary in nature and doesn't reflect the percentage of differentiation or the amount of differentiation. It only reflects that if a cell has a higher value of pseudotime, it just means that it's further along the biological process compared to a cell in the same biological process, which has a lower value of pseudotime. Okay, so how you perform trajectory inference. So there is a checklist or like a bunch of questions that you should ask before actually starting trajectory inference analysis, because any data set can be subjected to trajectory inference, but it might not yield any relevant results. So the first question is that whether your experiment is about a biological process. Now, some examples of biological process include differentiation. For example, in the case of bone marrow samples, where you have hematopoietic differentiation. So these kind of experiments, they are like single snapshot of a dynamic process that we just saw in the first slide. Or you can have the multiple snapshots of a dynamic process, which can be the case when you are sequencing development in the case of embryonic development. Or you can have disease progression where you have multiple snapshots along the experimental time, and the cells, they show changes or the dynamic expression changes by responding to some stimuli or some disease. And the examples include the development of cancer tissue. Now, the second requirement is whether your experiment has intermediate cell states. Now, what do you mean by intermediate cell states? So these are stages where a cell is not clearly expressing certain kind of markers or the cell type markers, but they are expressing some parts of it or not clearly expressing. So it's hard to put them in different kinds of, or hard to provide them labels, but it's very hard to consider them as dead cells. So unless you are putting some very stringent filters, they will end up in your data set, and then you might not be able to provide them cell type labels confidently. So these represent the intermediate cell states. So they are not fully functional, or not coming from or cannot be assigned to a cell type with confidence, but they still are expressing some kind of markers which are related to some specialized cells. So what happens is, in the cases where you don't have these cells, most of the algorithms, what they will do, they will try to learn disconnected trajectories. Now, disconnected trajectory means that it basically implies that you have more than one biological process in your data set. So you will have two different strings. So if we look at this example where we have this linear trajectory starting from this point and going until this point, so this is a continuous process and this is a linear trajectory. It's showing you a clear gradient where the stem cells are differentiating into more specialized cells. But let's say if you perform some kind of stringent filtering and or your cells were delicate and most of them died during the sequencing and you removed them. So you ended up having a data set like this where you have spaces in the middle. So this is not an ideal case for the trajectory inference. Rather, you should choose to do clustering here and try to infer biological insights from that point. Because if you will run trajectory inference algorithms on these kind of data sets, ideally in the default settings, most of the methods will try to learn disconnected trajectories over this data set. But if this represents a single biological process, for example, if this is coming from a single sample or a single snapshot of the biological process, then it might confound your analysis. Okay, so the next thing is, do you have information about the root cells? So the root cells are basically progenitor cells or the stem cell-like cells. Not always, but in most of the cases. So let's consider an example of hematopoietic stem cell differentiation. Now, in these, the HSC cells, they are the root cells. And root cells are typically given a pseudotime value of 0. So the idea of the root cells is that they represent the space where the trajectory starts or the biological process starts. And the progress of other cells is related to these cells. So how can you define these root cells? Now, there are different methods which you can use, but basically you can divide them into two categories. So you can either define them at the experimental level by sequencing or tagging your progenitor cells or sorting cells based upon the progenitor cell surface markers, or you can correlate your experimental design with experimental capture time. Or let's say if your experimental design did not allow you to tag them before or did not allow you to define root cells. So once you have actually performed the sequencing and you have the data, you can look for cells which are expressing the markers for the progenitor or stem cell-like genes. And then you can put those cells as the root cells. So we will see that in the demonstration, that why it's necessary and what happens when you assign cells as the root cells. Okay, so the method that we typically use and the method that we are going to see for this demonstration is Monocle3, which is maintained by the Trapnell Lab at the University of Washington. So this was the kind of a pioneer method which introduced the notion of pseudotime. It's a graph-based algorithm supporting all topologies. By that, what we mean that it can infer the linear trajectories or trajectories which represent some divergence or differentiation or complex trajectories where you have cyclical differentiation. And then it offers various data pre-processing steps, which includes normalization, batch correction, and dimension reductions. Since you have already seen that, we will skip these two, and we will directly focus on how Monocle3 works. So the Monocle3, just like any other single cell trajectory or any other single cell data analysis toolkit, it typically starts by representing the cells in a latent space. So these first two steps, you have already seen it with Martha. So we will start from this point. So once you have all the cells in a latent space, what you do is you learn a PQ graph. And a PQ graph is a specific implementation of a minimum spanning tree. And once you have this graph learned over the cells. So it's like putting a string through all the cells. You project each of the cell onto this graph. And once that is done, then you have to define where your root cells are, because that is the point from where the pseudotime is calculated. So you define based upon the known biology that where the starting cells are, and then a geodesic distance is calculated from the root cells to each of the cells projection on the graph. And this value is basically called as pseudotime. Now, this pseudotime is different for different kinds of methods, but for Monocle3, it's just a simple geodesic distance from the root cells to the other cells projection on the PQ graph. Now for the demonstration, let's first have a look at which kind of data set we will be looking into. So the data set that we will see come from this paper, and it's about hematopoietic stem cell differentiation. So this has CD34 positive cells, which are the HSPCs, which is hematopoietic stem cell and progenitor cells. So collectively, these are all stem cell-like cells, but some of them have already committed to some specialized lineages. For example, in this data set, we have the pure hematopoietic stem cells. We also have common myeloid progenitors, which will lead to the myeloid lineage-like cells, which includes the erythrocytes. And then we have the common lymphoid progenitors as well. So for this data set, these cells come from 3 different donors and they are healthy, but we will only look at the data set from this first donor. Okay, so as you have already seen how to perform the basic analysis, so we will start from the point that where we have already calculated the UMAP, and we have the cells represented in the latent subspace with, of course, performing basic QC and performing the dimension reduction and selection of highly variable genes. So this UMAP is already made for this demonstration, and we will start from this, and we will first learn the trajectory over this UMAP and try to see how the pseudotime is inferred, and then we will compare that whether the trajectory that we have learned is actually representing a biological process or not. Okay, so before we actually start doing the analysis, let's first revisit our checklist. So is this experiment about a biological process? And the answer is yes, because this has differentiating cells from the bone marrow. Does the experiment capture intermediate cell states? And the answer is yes, because all of these cells are the HSPC cells, and some of them are lineage committed. And what about the priors? So yes, we do know that we have the hematopoietic stem cells, which are pure stem cell-like cells in our data set. So we will put them as the root cells or the starting points for the trajectory. So without further ado, let's get started with the OmicsBox. Okay, so since you already have some idea about the interface of the OmicsBox from the previous presentation, I'll just quickly go through. So this is the result from Seurat clustering. What you have here is each of the cell, and their barcode is represented here. Then you have the cluster, which is assigned by Seurat to them. And then we have other information about the features and the count. So then this is the UMAP that we just saw. So we have the hematopoietic stem cells represented in orange, which are here. And then we have more lineage committed HSCs. For example, these red ones, these are early erythrocytes. So they are coming from HSCs to EMPs, which are erythromyeloid megakaryocyte progenitors, and they are differentiating into early erythrocytes. And similarly, we have this another lineage going into other directions. So ideally, when we will learn the trajectory, we should see a divergence here, starting from these HSCs going to 2 different parts. Now, to initiate the trajectory inference, you go to the side panel options of the Seurat results. So you click on the trajectory inference, and this will open the trajectory inference analysis wizard with Monocle3 in OmicsBox. So the first thing that you do is you select the root cells. So this is one of the most important step, because if you change or if you incorrectly identify the root cells, then your pseudotime ordering will be different. So since we see that we have the hematopoietic stem cells, we select hematopoietic stem cells as the starting cells. But let's say if you do not have them and your trajectory only has, your dataset only has cells which starts from the multipotent progenitors, you can also select them as the starting cells. But since we have them represented in orange, we will choose them as the starting cells. Next, we have to choose the transformations that we want to apply on the data. Now that this data is already processed with Surat, so we will not perform normalization again or the PCA again. We will directly work with the normalized counts. So we will skip them. But if you want to reanalyze them, reanalyze and start with the raw counts, you can select the raw counts, and then you have different options to do the normalization again from the raw counts. And then you have to select the embeddings. So we can see that it's already represented in the subspace of the Surat's UMAP. So we will just use the same embeddings, but feel free to change if you want to recalculate the UMAP, or maybe you have performed some kind of filtering over the cells. So you might want to recalculate the UMAP. But for now, we will just choose the Surat's UMAP. Next, we have to select the cluster method. Now, why do we have clusters in the trajectory? So the concept of clusters, which is typically done in Surat and in Monocle3, they are very different. So the clusters in Monocle3, they represent stable stages of the cells. So the likelihood of capturing a cell which is more stable and is more functional or is not an intermediate is very high. So those cells, they are represented by a cluster. And since we can change different kind of, we have different kind of options here, but we will go with Surat because we already have the clusters represented and we are confident that this is a biological process, so we don't need to recalculate the clusters. And then we have a bunch of trajectory control parameters, and we will see them at the end, what they are and how they change the trajectory. But let's first make our first trajectory. So upon hitting run, it will compute the trajectory, but I have already calculated it for you. So this is the trajectory for the same UMAP, and you can see these cells are colored by the same cell types. So we see that the trajectory starts from the point where the hematopoietic stem cells are, because we explicitly told Monocle3 that, okay, these HSCs, they are the root cells, so please start the trajectory from there. So the calculation of pseudotime happens from there, and we will see how the pseudotime looks in a second. To do that, you go to the side panel options, and now in the My Classification options, you have option of the pseudotime. So when you click on pseudotime, you have all the cells colored by a smooth gradient of the pseudotime. So these represent the pseudotime value of 0, as you can see here. So these are the hematopoietic stem cells. And as the cells are differentiating into more specialized cells, you have higher values of pseudotime. Now the trajectory analysis results also gives you a similar table, just like the Surat, but instead of the clusters, what you have is pseudotime for all the cells. We can rearrange it. So we can see that these are the root cells, and each of them have clusters. If you decided to go and choose the clusters from Surat, they will correspond to the same clusters. And then you also have partitions. Now, what are partitions? So as I mentioned while discussing about the intermediate cell states, if you are missing those, or you have naturally many different biological processes and you are learning disconnected trajectories, so typically each of the biological process will be assigned as a partition. And you can understand if you have, let's say, 2 disconnected trajectories, so you will have 2 different partitions. Now, of course, there are different different parameters which you can apply and explicitly tell Monocle3 that, okay, I'm 100% sure that I only have one biological process. So do not learn disconnected trajectories, rather connect my partitions as well. That, like, you can do that as well. But for that, you need to first understand that whether your data is actually representing it. Or you can perform clustering and then try to separate out biological processes and then learn trajectory on the different subsamples. So we will see that also, how you can change that. So, okay. So as we see that these two, they represent the actual hematopoietic differentiation of the HSPCs. Now you can also color this by different things. For example, I can color them by gene expression. So let's try to look at this gene and see its expression. So this is ABP gene. We see that it's expressed in the HSCs, and then it's not expressed anywhere in the UMAP. This is also one of the marker genes for the hematopoietic stem cells. Now, let's see some advanced parameters of the trajectory. So for that, let me first clear up some space and color them by the cell types again. So we go to my classifications and then cell type. Okay. And we can close this one. So I've already made the results for you. Let me open them one by one. Okay, so that's one, and that's two. Okay, let's first see these. Okay, so we go to the trajectory analysis and let's go to the last page where we have the option to control. Okay, so now we have the control parameters. So this is the trajectory that we learned with these parameters. Now, in this case, you see it's a more detailed version of the trajectory where you have more nuanced version of the graph learning. And that happens when you increase the number of centers. So you will have more detailed version of the PQ graph. Then we can cancel it. And then we see this one, let's say. Okay, so what this represents is cyclical structures. So you can tell the monocle, okay, I may have some cyclical trajectories or some patterns which are oscillatory in nature. So try to look for them. So for example, if you enable that parameters, what you end up having is the cyclical structures if they exist in your data set. For example, by default, you learn a trajectory like this, but then in the case where you have that kind of expression patterns, then it will try to learn these circular trajectories. Okay. And then what next we have is the pruning of the graph. So you can also decide to remove branches or the length of the graph, which does not meet a minimum criteria. So for example, if you remove a lot of them, for example, let's say if you set it to 5 and you prune the graph, then you will end up having a main trajectory, but not the branches. And then let's see the last one. Okay, so this is the case for the disconnected trajectories. So this happens when you have two different biological processes. So what Monocle3 implies that this is an independent biological process, and this is also an independent biological process. And one interesting thing to note here is if you color this by pseudotime, you will see that it only colors or it only assigns the pseudotime value to the partition 1, not to the other partitions. And the other partitions are given a pseudotime value of infinity, meaning that they come from a different biological process. But of course, if you are 100% sure, like we are, we can tell Monocle3, okay, these are coming from the same biological process. So please try to put them in one continuous trajectory. And in that case, it will draw this line and try to put them in one single trajectory. Okay, so now we have seen that how we can infer trajectories and what parameters we can adjust. Now we will see how you can infer biologically relevant insights. Now this is basically a pretty big data set, so we will not use all of it. We will first subset it. To do that, you can select on lasso. And let's try to select cells which are from this lineage. So what we are selecting is the hematopoietic stem cells. And then we have the EMPs, which are erythromegakaryocyte progenitor cells. We have early erythrocytes, and we have the progenitor megakaryocytes. Once we have them, if you see, they are now highlighted in the trajectory results, so we can right-click and extract the counts. This might take a minute or so, depending upon the size of the datasets, and then you can reperform clustering and reperform trajectory analysis. Okay, so we will not perform that. I've already done it for you, so we can directly see how it looks. So let's go to the subset of the trajectory. We try to make it, and let's hide this. And okay, let me color this by my classification and cell types. So what we have here is a subset of this main trajectory, because what we did, we selected the cells, we extracted the counts, we reperformed the clustering, and then we reperformed the trajectory analysis, just the steps that we saw in the previous steps. So what you have is the hematopoietic stem cells. Now they are represented in green. Then you have the erythromegakaryocyte progenitors, now represented in red. And then you have these differentiated or like lineage committed HSPCs. So we will work with this. This is a classic example of a bifurcation trajectory where you have one cell type differentiating or diverging into two different specialized stages. So, okay, so let's try to look at the expression patterns first. So to do that, let me go to the side panel options and let's look at the gene expression. So let's look at GATA1, which is one of the expression markers for the early erythrocytes. So we can see that this is not expressed in the HSCs. Rather, when the cell becomes more lineage committed, this gene is getting expressed. Now, there is a way which you can use to see the expression trend of this gene along the pseudotimes. To do that, you go to charts and you look at the expression trends of this gene. So we can write GATA1 here. And then we try to color it by cell type, and then we run it. So let's open them side by side. Okay, so we clearly see that when the cells differentiate from HSCs to more specialized cells, we have this expression marker going up along the pseudotime. So before you actually start looking into the novel things, the idea is to first confirm the known biology. So this is a very well-known marker for the early erythrocytes. And also we can see it clearly in this expression trend of this gene. As the cell becomes more lineage committed, we see the expression going up along with the pseudotime and also in the UMAP. Now, once you have confirmed few of the markers that you already know that they exist or should exist in your data set, you can start looking into novel things. Now, how can you find those genes which are having these kind of trends, like changing along pseudotime, or they're expressing at certain places along the trajectory? To do that, we can perform differential expression, or we can choose to do autocorrelation. So this is the autocorrelation wizard, but let's first understand a little bit of theory behind it. Okay, so autocorrelation, the entire name is spatial autocorrelation. So it's implemented in the Monocle3. So, and it computes a Moran's I for each of the gene. So it evaluates how a given element is similar to its neighboring elements. And in our case, our elements are cells. And it tests that whether a cell is expressing a particular gene and how the expression is compared to the neighboring cells. So this Moran's I, this basically ranges from -1 to +1. And you can have these three kind of patterns. So if you have a value which is close to the -1, what you will end up having a case where a single cell is expressing certain gene and all of its neighbors are not expressing that gene. Or you can have perfect randomness where the gene is expressed everywhere in the UMAP, or maybe not expressed everywhere, but it's purely random. Or you can have a case where you have perfect clusters. So it means that a cell and all of its neighboring cells are expressing its gene, or maybe not expressing this gene. So we will see that in our analysis, how that looks. So typically this is used when you want to look at the gene expression and you want to find out genes which are significantly expressed in certain branches. And mostly you are interested in the cases where you have Moran's I value, which is close to the 1. For example, we see in this case, the Moran's I value is 0.8. And we clearly see that this gene is only expressed in one branch, not in the other branch. So this is a scenario which shows you a perfect cluster. And then we can have a look at the perfect dispersion. So here you can see that the gene is expressed in certain cells, but not in its neighboring cells. So now we can perform that with our data. So we have different options for the autocorrelation as well. The first option is the neighborhood graph. So Monocle offers two options to perform this analysis. Either you can use the trajectory graph, or you can compute a KNN graph over the UMAP again to perform this Moran's I analysis. In our case, we are interested in the principal graph. And then you select the hypothesis and the family. In our case, this is single cell data, so we will choose negative binomial distribution, but you can choose, depending upon your data set, the other ones as well. And we can run it. So instead of running this, I already have the results stored, so we can directly go and see how the results are. Okay, so these are the results from the Moran's I analysis. So on the first column, we have the tags, then we have the genes, because this analysis is done per gene. And then we have the gene name, and then the values of the Moran's I. So we have the Moran's I, which ranges from -1 to +1. We have Moran's test statistics. And for each of the test statistic, what we have is a p-value and adjusted p-value. And then in the tag column, you see some are significant, some are not significant. But this is something which you can adjust on the side panel options by going to update tags. So you can adjust that which are the genes that you want to be tagged as significant. So you can actually select whether you want to filter them based on the Moran's I or statistic, whether you want to get the genes which are having a Moran's I value which is close to the 1 or 0 or -1. And then you can set the significance criteria, whether you want adjusted p-value or p-value, and then you can define a threshold. Once you do that, these tags will be updated. So let's try to look at these genes which are significant. Okay, let's first update the tags and then we will see. So we want all the genes to be significant that have a test statistic value greater than 1 and a p-value, let's say, smaller than 0.01. Okay, so when we run it, most tags are updated. You won't see them here because these are already updated. Okay, so as we see that most of the genes which are marked as significant, of course, they're mitochondrial because we are looking at the differentiation and this requires a lot of energy. So it's obvious that these genes will be expressed. But let's try to look at some genes which have the negative value of the Moran's test statistic. Okay, so one of them is SYNC. This has a value fairly close to 0 and negative. So, okay, let's try to look at the expression of this particular gene. So we go back to the trajectory and yeah, let's try to color it with here. So the name is SYNC. And let's remove the GATA1 expression. And let's change the higher mid expression value to red as well to see it clearly. Okay, so what we see here is that this SYNC gene has a negative value of Moran's I, which is very close to the 0. So we might expect some randomness, but still it has to express a dispersion pattern. So we see that this gene is expressed in certain cells, but you will not find the neighboring cells expressing this gene. Now let's try to look at a gene which has a very high value of Moran's test statistic or Moran's I. So let's look at SPINK2 gene. And we go to the trajectory. And let's try to look at SPINK8. Or was it SPINK2, I think? Okay. SPINK. Okay, so this gene is not expressed. This is SPINK8-2. Maybe we filtered it during our analysis. So let's try to look at the GP9 gene. Okay. So this is a GP9 gene, and this is a known marker for the progenitor megakaryocyte. So we can see that it's expressed in a certain location and in all the cells which are neighboring that location, in that location, but not in the other cells. So it has a relatively high value of the Moran's I. And let's try to look at the expression of this particular gene. So we can go to the charts again, and we can look at the expression trends, and we can write GP9. And let's try to look at the expression. Okay, so we can easily see that the expression here. I'm going to save it. So it's expressed in progenitor megakaryocyte cells here, and we see that it's it has no expression in almost all of the cells, but it's only expressed in the certain location. So that's why we see a upward pattern in the progenitor megakaryocytes. Okay, so this is how you can perform the autocorrelation analysis, and you can change tags for the different genes. And once you have tagged them based upon your needs, what you can do is you can perform Fisher exact X. So you can supply the annotations for your genes, and then you can try to look for the signatures for them. Okay, so let's go back to the presentation. So as we have already seen it, and some final thoughts. So any data can be subjected to trajectory inference, but you have to understand and ask yourself that whether your data set really represents a biological process and whether you are capturing intermediate cell states. Then you have to validate the trajectory with the known biology. So before you actually start finding novel things or try to look at the expression patterns which are novel, you should first confirm that whether your data is representing the known biology or not. And then lastly, this trajectory analysis or single cell data analysis in general, it's a very nonlinear approach to data analysis. So you have to constantly redo the analysis and check the analysis for the patterns from your domain knowledge or your information about the experiment, because there is no one size fits all for the single cell data. And lastly, I thank you for the time, and thank you, BioVam, for the opportunity. And that concludes my presentation.
Thank you for watching. Learn more about OmicsBox with our other videos.